last physiology lecture, we're having trouble getting it posted. I don't know something's gone AWOL with the system, so I'm hoping to get done with it. All right, so codominance. We did some of Mendel's standard monohybrid problems. What is Mendel's standard phenotypic ratio for a heterozygous cross and a monohybrid problem? You remember? Phenotypic. Do you remember anything from?
3 to 1.
Very good. It's been a while back, so we're trying to review a little bit. What about the standard genotypic ratio for that same cross?
1 to 1.
That's it. Very good. So those definitely are test questions because we said Mendel's stuff is predictable, and as we get into the dihybrids today, they'll be even more predictable. We worked some sex-linked traits the other day with color blindness and hemophilia, and we saw how those connected. And if we say sex-linked, we're typically talking about what chromosome? Where is it carried?
X.
On the X. We identified a couple of things on the Y, but not a whole lot. One of them was the testis determining factor, and the other thing was hairy ears. You know, there's some things we'll never forget. Now, whether it's real pertinent in life, I don't know. The trivial stuff we'll always remember. So today, we want to start on page 79 and look at Mendel's dihybrid crosses. In Mendel's dihybrid cross, we're still going to do a big T for tall, a little t for dwarf. We'll do a big Y for yellow pods, and we'll do a little y for green pods. So we're working with two traits. It's a dihybrid, not a monohybrid. There's one little trick you have to do here before we put it in a Punnett square. If you don't do this one trick, it'll never work out. But you should be able to identify it. Mendel always liked to cross two homozygous versions of this. So he'd cross a big T, big Y with a little t, little y. What combinations of these gametes can this parent offer? There's only two sets there, so what combination can that parent offer?
Big T, big Y.
Very good. Big T, big Y, and that's it. Everything else repetitive. And what you'll see a lot in this is that if it's repetitive, there's no need of writing it down again. What about this parent?
Little T, little Y.
That's it. So all the offspring from these two parents would be big T, big Y, little y. Notice I keep the uppercase letters first. I keep like letters together. That reduces confusion. You don't want to look at a Punnett square box and it be big T, little y, big Y, little t. You don't, I mean, that's just a mess. This is a mess enough, so let's keep it neat and more organized, easier to understand. So this is Mendel's F1. He liked to cross his F2. That's this one. So if you cross two of those, big T, big Y, little y, what combinations can this parent offer? Big T what?
Big T or little t?
Now what I do combinations?
Big T, big Y.
Big T, big Y.
Little t, little y.
Let's stay with the big T first. We can do it in the big T, little y. Now we go with the little t.
Little T, big Y.
And
Little t, little y.
there you go. Now, what you did there, how many's had algebra? Usually people just shut down brain when I say algebra. I don't blame them. Probably reminds you of Mr. Holden, and that would shut me down too. What you did here was in algebra, you learned a full method. That's all this is. It's the full method of algebra. And if you say, well, I didn't pass algebra, then this is not going to work out very well, is it? And you was asking Mr. Holden, what on earth would I ever use this for, right? Zoology. There you go. Question? All All right, so what you did,
Is that a little t, big Y, little t, little y on the very bottom? Is
Yes.
the only big one a Y? on
we
the
have a
left?
big T, big Y, right here. Big T, little y, right here. Little t, big Y, and little t, little y. I'm gonna do the same with this parent. Big T, big Y. Big T, little y. Little t, big Y, and little t, little y. Now, you know, in representing traits, you can represent a trait with anything you want to. I mean, it may be big T for tall, but you could use a Q if you wanted to. But what you want to do is write it down, and you want to try to use letters that if you get on a roll, they don't start looking the same. You know, these T's, notice I put a kink in the little T. I'll make it with a hook or something, because when I get going real fast, that big T may get crossed right there. Is that a big T or a little t? Well, it doesn't have a crook on the end of it. See what I'm saying? The W's, you think W's start looking the same after a while?
Yeah.
Which one's which? My little W's have a hook on them. So use things that you can certainly separate, so when you're done with Punnett square and you're going back and looking at it and say, what was that really supposed to be? That's not a good situation to be in. That's where your confusion comes in. So these things really require you to take your time, just to make sure you don't make the little mistakes that'll catch up with you. You know, have a knowledge of it, to, oh man, it's rolling now. You want to get it done, and when you get done, what the Sam Hill did I just write down? That's not what you want. All right, so we're going to take and create a dihybrid Punnett square. Just be glad we're not doing a trihybrid. We don't want 64 squares up here. So what we did, we have all the gametes, combination of gametes necessary to use on this square. We have a big T, big Y, big T, little y, little t, big Y, and a little t, little y. The other parent was the same, wasn't it? Now, if you're working one of these, and we're going to work some of these today as we flow this lecture into lab and work through that worksheet that I handed out. In fact, we may work through that one and part of another. If you are setting up a Punnett square and they give you the parents, let's say we have these same two parents right here. We've got this cross right here. Big T, little t, big Y, little y, cross with itself basically. And you say, okay, put a big T, big Y, little t, little y. Does that even look right?
No.
Well, on this side, well, let's just put a big T and a little t and a big Y and a little y. What do you think that's going to turn out to? See, in a dihybrid, you have to make sure you have combinations of two traits to represent each square. This is just going to represent the height of the plant. Down here, we're just going to get pod color. It's not going to do us any good. So if it's a dihybrid and you immediately see one trait represented per square, just erase it. You've already messed up. Back it up. That's the most common mistake made in dihybrids, is not doing the full method. You have to do this. Okay? Now, if we were to set this up in stages, like the first thing, you want to show cross. Well, let me back up. You want to represent traits. And a lot of times the problems will do that for you. Okay? Well, and number two, show cross. And sometimes the problems will do that for you. That's kind of the easy stuff. Step three is show gametes. And we'll just write full method in parentheses. That's the step that's skipped 90% of the time in dihybrids. Step four is the Punnett square. And step 5, I want you to just circle your answer so I'll know which one it is. No telling what you'll come up with at the end, so I wanna know what you think the answer is. These things are difficult enough to grade without not being organized. So, believe me, we've seen a bunch of stuff come through the office before. It's like, I'm not sure what they're saying here. So, if you'll follow these steps on dihybrid problems, you won't miss a lick. Now let's start putting this together. And y'all can do it on your own paper there. And the one in the book is, it may be identical. Now, it should look something like that when you're done with it. If that was only the finished product, it would be fine. Now, if you're writing those in real fast, you can see how this can become confusing if you don't take your time and make your letters what they're supposed to be. Now here's the question Mendel had, and he answered it. We're gonna learn it because this is predictable stuff. He wanted to know what the genotypic ratio and the phenotypic ratio of this combination is gonna be. So the phenotypic ratio, what are we looking for when we look for phenotype? What they're looking like. I mean, if we took that out of the square and made it alive, what would it look like?
It'd be a tall yellow plant.
It'd be a tall plant with yellow pods. That's exactly right. If we took this one out and made it alive, what would it look like?
Be a dwarf.
Dwarf with yellow pods, wouldn't it? That's what we're looking at. So let's look at this one right here. We know this is tall and yellow. So let's look through each square here. Is this one tall and yellow physically? What about this one? What about this one? This one? Nope, that's got some green pods, don't it? This one? This one? This one? This one? This one?
Yeah.
We have 9 that are tall with yellow pods. Now it's important that you write tall and yellow. When you write your number, write what that number represents. 9 of them, tall, yellow. All right, now let's pick one we haven't looked at. What's this one gonna be? Tall and green. What's this one? Tall and green. That's not tall, not tall, not tall, not there. What's this one? So we have 3, tall and green. What's this one? Dwarf and yellow. What's this one? Dwarf and yellow. That'd be awful, wouldn't it, be dwarf and yellow? That dwarf and yellow too? Dwarf and yellow. There's only one worse than dwarf and yellow. Dwarf and green. One that's dwarf and green. I will ask you on the test, this 9-3-3-1 ratio. We're gonna work a couple problems on this paper that may not exactly come out 9-3-3-1, but the genotype will come out the same. So the question will be something like, what's Mendel's standard heterozygous dihybrid phenotypic ratio. Think about that. Mendel's standard dihybrid heterozygous phenotypic ratio. Standard, in other words, he's proved it. This is predictable. Dihybrid, 2 traits. Heterozygous, those traits are all different. Phenotypic, we're looking for physical makeup. That's what this is, right? 9331. Now we want to identify the genotypic ratio. The genotypic ratio is actual genetic makeup. So can you see there might be quite a bit of this? Because is there another one on this whole chart that looks like that one genetically? No. Is there another one on the whole chart that looks like that one genetically? How about that one genetically? Well, so there's a lot of singles in there that's just like, I mean, there's nothing else like them. So here's your genotypic ratio, then we're going to fill it in. 1, 2, 1, 2, 4, 2, 1, 2, 1. Again, being organized is going to help you with this. Notice the way we took big T, big Y, big T, little y, and we went down using this one, this one, this one, this one. Notice that? We did it across the top, we did it down the side exactly that way. We can fill the genotype out by this. 1, 2, 1, 2, 4, 2, 1, 2, 1. We have 1 big T, big Y, big Y. We have 2 big T, big T's, big Y, little y's. We have 1 big T, 2 little y's. We have 2 that's big T, big Y, and 4 that's just like the parents. And 2 that's big T, little Y, little y. We have 1 that's little t, big Y, big Y. 2 that's little t, big Y, little y, and 2 little t, little y, little y, or 1, excuse me. So there it is. Now, a question we're going to work here in just a minute on this handout is going to ask about, it's a little bit odd, this, the genotype is going to be exactly this. The phenotype will be off from what seems predictable, because it's going to be asking about color of this flower. And they'll tell you that to get a purple flower, you have to have the pigment or the gene for the color purple, and you have to have an activator gene that turns on the color purple. I think it's number 6. What was number 6 on that? Not it. Try 7.
2 sets of factors called flower color and sweet peas.
Yeah, sweet peas. Yeah, that's what we're dealing with. Let me see that. Number 7. There's 2 sets of factors called complementary, responsible for flower color and sweet peas. These genes may be represented by, see, and they're represented for you. P for purple, and E for the activator. You could use an A for activator if you wanted to, but they use E for activator. And it's an activator for the color purple. These 2 dominant genes must be present to get purple color. It doesn't say they have to be homozygous dominant, they just has to be a big P and a big E. Any other combination is white color. If we have 2 big P's and no big E's, it's white. If we have 1 big P and no E's, it's white. If we have little P's and big E's, it's still white. We have to have a big P and a big E. So, first thing they give you in this problem is 2 white varieties. They have a little p, big E, crossed with big P, little e, little e. would you agree both of those are white according to the rules? Definitely white. Now, what combinations can this parent offer?
Big P, big E.
What combinations can this parent offer?
Big P, little E.
If we put them together, we got big P, little E, little e. What's the color of all their offspring? Purple. Every one of them's gonna be purple. Looks to me like you got a bunch of pee there, but it'll get worse once you work the big square. So what they're wanting to do is see what your genotype and phenotype ratio from this purple offspring is now. So you're gonna cross two of those. Now does it look familiar from what we just did? So we have big P, big E, right? Big P, little e, big E, and little p, little e for this parent. Big P, big E, big P, little e, little p, big E, little p, little e for that parent. So now we do a big 16 square. Some of y'all's working on it, some of you's not. You plug in this 16 square. I'm doing homework for you, so jump on this. I'll give you a few minutes to do that one. If I get out of your way, you're going to get a crick in your neck trying to look around me. That's how it should look, and then you fill it out. And then you tell me what you come up with for a phenotypic ratio, and then your genotypic ratio. Last one should be little p, little e.
Okay.
Doesn't look that way, but little p, little e. That's another reason why you take your time writing those. Is P is another letter that could eventually start looking the same. Now E's, you can certainly make them different. Make your standard print big E in a cursive little e. Some Heavener graduates in here. I thought I recognized some. You have Mr. Mead?
Yeah.
Yeah. He taught you a little bit of this, didn't he? Didn't y'all play with the Punnett square a little bit?
He tried to teach us.
In his latter years, he liked to visit a lot, though, didn't he?
Oh, yeah. We're pretty good at that. We kept him distracted.
Yeah, y'all caught on to that pretty quick, didn't you?
We're good at it.
I can only imagine. He bought right into it too, right?
Oh, yeah.
What year did y'all graduate?
2012.
Oh, y'all just graduated. All right, some of you have this and some of you's close. Now let's look at the phenotype first. Take the very first square, big P, big P, big E, big E. What color are those P's? They're purple. Now go through every square and find how many purple P's you have. Kind of like Peter Piper picked a peck of purple peas. How many purple peas did Peter Piper pick? How many we got here?
9.
We have 9. She's already counted them. Let's see if anybody agrees or disagrees with you.
I agree.
You agree? They're on board with you. I think you're right. So we have 9 purple peas. Now, unlike this problem over here, we had other options, didn't we? We had combinations tall and green, dwarf and yellow, dwarf and green. What's our other option?
White.
White. What's left? 7. 9 to 7 is your ratio on this one. Not exactly a predictable Mendel problem, but certainly enough information to work this one out. But when you first see the cross, you say, that looks like one of Mendel's standard crosses right there. I mean, that's why it's predictable, because you should start recognizing this stuff. So on your genotype, is it fitting our standard genotype?
Yeah.
Right to a T. 1, 2, 1, 2, 4, 2, 1, 2, 1. Right to a T. So far, so good? That's not too bad? Yeah, and that's probably one of the more difficult ones on here. So it's a lot easier when we work through it than with a little guidance. So I'll turn you loose on some here in just a minute. Back in your text, look on page 81. I want to hit a few terms here, do a little bit of explanation as we get over the next couple pages. On this particular page, on page 81, gene interaction. It says many genes have more than a single effect on organismal phenotypes. This is a phenomenon called pleiotropy. In other words, many genes have more than a single effect on the phenotype. Many genes involved in a particular phenotype. For example, I think we used eye color as an example. There's like 8 different genes involved in eye color. I think there's more than that involved in an Appaloosa horse. I mean, that's polygenic inheritance is another term you could use for that. An allele at one locus can mask or prevent the expression of an allele at another locus. And a locus is a point on a chromosome. So this allele up here tends to mask this allele down here. And they may be for different traits. This allele is dominant for this trait, yet it covers this trait. And this is called epistasis. Many cases are known in which a variation of many different genes can affect a single phenotype. So very close to our definition of pleiotropy, we have polygenic inheritance. Quantitative inheritance is a case where you get an intermediate combination of 2 parents. If you have a tall parent and a dwarf parent, you get a medium-sized child. That's what they're saying. And we all know that doesn't hold up 100% of the time, because you could have 2 dwarf parents and have a tall child. I mean, that can happen about 50% of the time. So, all right, flip over to the next page. Now, we've already covered some sex length, and that's what they're talking about here. We have a few that we will work on this sheet here in a minute. The term crossing over is on page 83. Where does crossing over occur? You should recognize that term back from chapter 3, test 1. Whoops, maybe we missed that one on test 1. Where?
Mitosis?
It was in
Or
meiosis.
meiosis.
Yeah, prophase 1 of meiosis, that's right. All right, so look on 84. Let's look at some terms here. Changes in chromosome numbers are called euploidy when there is an addition or deletion of whole sets of chromosomes. So addition or deletion of entire sets of chromosomes is called euploidy. That's not, especially in animals, this is not a good situation. That's lacking information. Lacking information is closer to a death sentence than too much information. Too much information causes problems, but it causes confusion, yet there's still life in a lot of cases. But whole sets, oh, that's really messed up. Now, it's aneuploidy when a single chromosome is added or subtracted from a set. A single chromosome. This would be a case in added or subtracted monosomic conditions. That would be N minus 1. In other words, if let's say N is supposed to be XX, but it's not, it's XO. O represents a missing chromosome. That's a monosomic condition. And that's exactly what they're talking about here. An additional set would be trisomy 21, where there's 3 chromosomes on set 21, Down syndrome. Or you can have 3 X's on one. You can have 2 X's and a Y. That's extra. We're going to go over some of these in a minute. They're not in the book, but I add them to it. So that is aneuploidy. The most common kind of euploidy is polyploidy. I know this is a lot of ploidy, but polyploidy, occurrence of 3 or more sets of chromosomes in an organism. Let's just say if this happens in animals in general, it doesn't, but animals don't like it. Animals do not like any kind of change, mutation. The genetics just, it typically rejects it. You know, and we hear of cases right and left of miscarriages and pregnancies. Let me tell you, a lot of those miscarriages and pregnancies are due to rejection of the body because of chromosomal defects or lack of or too much of. And it's the body not recognizing it as human, therefore rejecting the pregnancy. And as sad as those are, and physically and mentally they work on the mother, there's a reason for most of those. So, in plants, we encourage polyploidy in plants. You know, we're getting real close to gardening season with a week like this. It's in the 60s. Went down to the co-op day before yesterday, and they had 4 boxes of onions, maybe 5 boxes. They had 1 bunch left. I mean, the onions are gone. So people's you know, wait till they start putting out the tomato plants and the pepper plants. Then go down there. I ought to make this assignment because nobody ever goes. Just go down there and pick it doesn't matter, pick a tomato plant and pull the little plastic deal out and turn it over and read on the back. And it'll have a bunch of letters that really won't mean anything to you, but they're different kinds of viruses and diseases, and it'll say resistant. They have genetically altered this plant to be resistant to certain diseases that are most common to this plant. In other words, this plant's pretty tough. It's a no-brainer to stick it in the ground and pick a tomato off of it later. Geneticists and greenhouse owners have learned to make a plant that even the novice person could raise fruit from. I mean, that one greenhouse owner, when I used to teach botany, I'd do 2 field trips out there. I'd do one early in the season when he'd go through the seed planting process and transplanting process, and then I'd do one later where he actually would go in with a chemical that would stunt the growth of the plant. You say, what do you want to do that for? Oh, I just want to slow it down for a little bit. He said, if I slow it down for a little bit this way, it'll get bigger this way. And so when it gets bigger this way, it builds more chloroplast, which is the energy producing part of the plant. And he said, then when it grows, it won't be quite as tall as it could have been, but it'll be much stouter. it'll withstand not only more wind by being a little more of a dwarf plant, but it has more chlorophyll, it'll produce more energy and produce more fruit. Just by one little chemical at a certain stage in its life. So they've learned to do stuff like this, so non-green thumbs like myself can't just kill it as soon as I touch it. It has a chance for survival. And these greenhouse people, I mean, they're plant nuts is what they are. I mean, this is the way he talks to us when they look at these plants like their own children. He said, we want our plants to have a chance to survive. I mean, we don't just throw it out in the road so they get run over. And granted, most people, I mean, didn't y'all know how to plant a garden? You know a little bit about the soil, you need to do a soil test, you need to know where your pH is at, or do you know just to dig a hole and stick it in there?
Dig a hole and stick it in there.
There you go. And that, you're exactly the person that they're preparing their plants for.
Hey.
And if somebody knows, and if you do that, you'll learn more. There'll be things go wrong and you'll ask questions and you'll learn more. But it's amazing what we've done with plants by polyploiding these genetics. You know, we raise the crops we raise today in huge numbers because we've genetically altered a lot of these. Now, there are some countries that will not let us export wheat and some different grains because they don't want genetically altered grains. What does it do to people? We can't see it's done absolutely anything except increase crop numbers and lessen disease and lessen overall expenses, because if you have to use chemicals to fight things off and pests and everything, I mean, the cost of the crop goes up, and you don't want to eat this stuff that has chemicals on it anyway, or at least you'd rather have it organic, wouldn't you? As close as possible. So that's where we're at with polyploiding. We do it on plants. It doesn't work for animals. It's just a messed up deal in any animal. But plants, we do it all the time. I mean, if you can take a single rose bush, and as this rose bush grows, it produces literally every color of rose flower known to man. Now that's genetically altering a bush. And they can do that. They have done that. But they can't take that bush and replicate itself. They have to go back to the beginning, kind of like the blue Indonesian chickens. To get that bush again, they have to go back and make that original cross. If they take parts of this bush and cross it, they might get a whole bush of yellow flowers or a whole bush of pink or a whole bush of red, but they won't get all the combination. So, all right, now, the cause of some of this. The term non-disjunction. I'm going to explain this here in just a second. And below that is monosomic, N-1. I use the term trisomic, that's N+1. That's an extra chromosome on a set. And then they use an example of trisomy 21, Down syndrome. You know, Down syndrome falls in a category of what a lot of books refer to as a 40-40 syndrome. There's a 40% greater chance of women over 40 of producing children with this kind of syndrome. And what does that tell us? What does that mean? You shouldn't have kids over 40? If that's what it means, somebody should have told that to my wife. She had 2 over 40. I thought she was crazy. Now I have to raise them. They're killing me, both of them. I don't have the energy. Just when I catch them, I eventually give them a whooping, but I just can't catch them anymore. What this is saying is that females are born with all the eggs. They're born with all the eggs they'll ever have. Okay? Just like any other cell of the female body, with age, replication of those cells may not be as good. Okay? So, non-disjunction could occur, which causes these problems. Now, you can't say that on the male side of it because sperm is regenerated literally every 60 days throughout a man's life. She's born with all the eggs she has. Something doesn't seem fair about this, so if she decides to do all kinds of drugs and take all kinds of radiation treatments, whether she needs them or not, and fry her eggs, they're fried for life. Now, guys can go out, I'm not advocating this, you should consult your physician before you do any of this, there's my disclaimer. Guys can go out and kill sperm right and left and maim it and throw marijuana on it and cocaine and it just decide to quit cold turkey, and 60 days later they can produce normal sperm again. Doesn't seem right either, but that's what this is saying. With age, your cells don't replicate like they did when you were 20 years younger. So the prime years for reproduction in humans is typically 20 to 30. 35 is not uncommon these days. Over 40 is not recommended. That's just the way it goes. Now, look over on page 85. I'm going to come back to non-disjunction in just a second. The left-hand side of 85 says a portion of a chromosome may be reversed, placing the linear arrangement of genes in reverse order. This is called inversion. Non-homologous chromosomes, these are not sisters, okay? They're nothing alike. May exchange sections, called translocation. That's not normal, that's not good. If sister chromosomes exchange sections, that's what happens in crossing over, that's all right. So translocation's not good. If there's entire blocks of genes lost, that's deletion, that's lacking information. That's not good either. And then sometimes we replicate these things called duplication, causing serious developmental defects, or an extra section of a chromosome may attach to a normal chromosome and duplicate itself. All these causes some serious problems. Let me give you one example. In chromosome set 5, if there is a deletion of portion of set 5, there's a syndrome that they named Cry-Du-Cat syndrome. It's spelled CRI/DU/CHAT. It sounds like Cry-Du-Chat, but it's called Cry-Du-Cat syndrome. What this is a small, when this child is born, this child's head is disproportionately large to its body. They're nearly positive there's some obvious mental retardation involved in this, but when this child cries, the child's cry sounds like the meow of a cat, and typically will not live past a year, maybe 2 at the most, and they die. So that indicates a lot of nerve tissue damage, because there's a lot of those situations where in a lot of cases there's too much information and it confuses the nervous system, and when your automatic system like breathing, heart rate, all this is supposed to work on its own and your brain forgets to do it, you're dead. I mean, that's just what happens. And that even happens in normal children from time to time. SIDS, sudden infant death syndrome, that's basically what happens there, is the medulla forgets to breathe. Now, we blame other things for causing that. In some cases it may be, but ultimately your medulla just forgets. It's not a good thing to forget. Now, with that said, let's look at 4 chromosomal anomalies that occur on the sex chromosomes. This is not in your book, so you'll want to take notes on this. The first one, XO, is a monosomic condition, and it's called Turner's syndrome. Turner syndrome. This is a female, and if we put a description to her, and these textbook descriptions are not always real life descriptions. They may be missing some of these descriptions, or they may actually have more. So the textbook description would be a female that's short in stature. Now don't go out in the parking lot and look at everybody that's short and think, must have Turner syndrome. There's a lot of normal short people in the world, okay? And when they say short in stature, I've actually had students come through class and they had to tell me they had this syndrome. And they weren't abnormally short, probably taller than some of y'all in here. So why they call it short in stature, I'm not sure who we're comparing this to. Jolly Green Giant? I don't know. But short in stature, typically below normal intelligence, which didn't fit this person because she already had 1 degree from major university, come back to go through a nursing program. I don't think you do that being below normal intelligence. A webbed neck. Yeah, explain that one, huh? Skin extending from the neck out, maybe a shorter neck that looks like more skin extending out to the shoulders. This person did not have that. Now, you could Google up this at any time on your phone, and they're going to show you pictures of this, and they'll probably show you a webbed neck picture. All right. So, can this person reproduce? Most books will say no. I'm not going to say she can't, because I think there's probably cases she can reproduce. Now, if I were to pick one thing that I thought was maybe different about this person in class, she didn't socialize very well. She wasn't good at socializing. She wanted to, but she wasn't good at socializing. A little bit introverted. It's hard to say that that's totally abnormal. There's a lot of people like that that's normal. Next one, XXY. XXY is Klinefelter's syndrome. Klinefelter's. Klinefelter syndrome. This is a man. This is a male. A male with an extra X. That extra X gives this male a little more estrogen than he needs. So this male may have underdeveloped male genitalia with breast development. Do you think he can reproduce? No. He cannot reproduce. He could be of normal intelligence, though. As far as surviving a normal life without reproduction, he probably can. Now, this person was identified in the Olympics not long ago. This person was in the Olympics running races against the women and won a lot of races against the women. Not sure how it came about, but somebody suspicioned something, so they run a genetics test on this person and found out that she was a he and disqualified him from all of her races. Did you get that? Now, if you're going to play in the Olympics, you're going to do a genetics test up front. So we're going to play fair. Well, let's think about this. Here we have a male running against women. Let's look at these female runners. I mean, some of y'all ran track in high school probably, and we've certainly watched some Olympic games. And I mean, these athletes, they're a lot of muscle and not much fat. Body fat's literally nothing, would you agree? Especially on these women runners. So if body fat's literally nothing, what do you think the muscles on their legs look like? Pretty profound. What about breast development on these women that are running? I mean, granted, a lot of breast size is built off of fat. If there's no body fat, then breast size is going to be small, okay? So they're machines. All right, now let's describe this guy. We already said he has breast development. What do you think the muscles of his legs look like? Now, if he shaved his legs, if he didn't shave his legs, he'd be an awful ugly guy, wouldn't he? Maybe ugly won't know it's an ugly guy. It's a little confusing. So we're watching the Olympics and say, man, that woman right there is faster than greased lightning. That's the ugliest woman I ever did see. Well, it wasn't a woman. It was him. So really, you can see where he could kind of get by with it. So he's ugly. We get by ugly. He's quite an athlete. Or she's quite, no, it's he, wasn't it? See how confusing this is? So anyway, now, we can apply this outside of humans. Let me get the pee-pee off the board. If we apply this outside of humans, let's look at calico cats. Any cat lovers in here? Anybody like cats? No cat lovers? Oh, okay. Everybody else is a good cat's a dead cat kind of person? You just don't care.
We have like them.
Oh, you better like something about them, though. Cats everywhere. And calico cats, you know, that's a unique combination of colors to get a calico kitty. They have to be on the X chromosome. So the female calico cat gets your combination of colors, and that's fine. That's female calico. To get a male calico, you don't like cats? It's just upset her terribly, didn't it? Have to have two X's in the Y. The Y gives us a male. We have to have the two X's to get the combination of colors. Has anybody ever had a calico cat? Have kittens?
No.
Not had kittens? But it is a female.
Yeah.
Well, that's a main point. If you ever have one to have kittens, if it comes out a calico color, it'll be a female. If it comes out a different color, it could be a female, but all your different colors are typically males. And it's like the old yellow tom. You ever seen the old yellow tomcat? You know, and that would be the father. Yeah, pees on everything, stinks like. And that's the father of all kittens for a 5-mile radius. Is that old yellow tomcat? Yeah. And then they get kind of a muckly, brindley looking color. It's not even pretty, but it's kind of got slashes of all the calico colors, but the background of it, it's just ugly. You got some of those too?
We got one in our yard that just comes around, doesn't leave.
One ugly cat. Yeah, that's it. Well, that's probably born from a calico mother. All right? So this cat here is a male calico, but it has Klinefelter syndrome, and that's a problem in cats. There's two problems with this. This male, number one, is sterile, and number two is mentally retarded. So I don't find that abnormal to most cats. I think most cats are slightly mentally retarded. I have one of those at home myself. That cat of mine will stand up. Top of the tail will come up to here on me. But it itself stands up that. We call it mini cougar. It looks like a cougar, and it's barely mini cougar. I mean, it's bigger than my dog. Put it that way. And can whoop the dog when he gets ready. So, in calico cats, to be a male calico, it's going to be sterile and mentally retarded. Most of these, because of the mental retardation, is usually lost during a pregnancy or stillborn. When they're born, they'll be born dead. Very few are born alive. Okay, so the cat's body is saying, oh, there's something wrong with this cat. They cannot survive society, so it's either stillborn or it's rejected in the pregnancy. So if you see a calico cat cross the road, you could win a quick bet and say, I bet that's a female. And then it'd be my luck, it'd stop, you'd run over it, so, oh, that's a mentally retarded male. I had a cat one time that, and it was kind of a Siamese cross cat. Boy, it's a mousing machine. She was well, let's just say she had a lot of kittens from a lot of different, she got around, okay? And I don't know why, I named her, this was her name from day one. I named her Stupid. That was her name, Stupid. You know how she died? She got run over by a car. Wasn't that stupid? I always knew when she was in heat, there'd be 27 tomcats I'd never seen before show up. I'd flush them out like quail. I had a ball. I learned how to shoot that way. But there was just way too many cats. But that's Klinefelters in cats. Let's look at the next one. This one's called meta female. Meta female. Say, well, 3 X's. What do you think when you see 3 X's? I'm thinking, what a woman. I'm really thinking way too much estrogen. Do they find anything abnormal with this person? What do you think? normal? You don't think she's normal? She could actually live her entire life and not know she's a male female. Normal intelligence, function properly in society, she can reproduce. There may be no reason to ever do a chromosome check to ever find out she has this. The only thing that's ever been documented, and it's not even printed anymore because they don't consider that normal, is that her 28-day cycle may be erratic. What's abnormal about that these days? Not that abnormal, is it? I mean, stress in life can cause erratic cycles. Illnesses, genetics. What if you're one of those athletes that's training all the time? They could go 2 or 3 years without a cycle because their body, see, the human body will focus on survival first and reproduction second. And if you spend so much energy in training for a triathlon, then your body's not going to worry about reproducing. So erratic cycles is not considered abnormal anymore. So there's your meta-female. Now the one you ladies have been waiting on here, the super male. I always leave it to last because it has the most interesting name. Like to get a hold of you a super male one of these days? No, I wouldn't either. Too much testosterone. So what do you think's wrong with him besides too much testosterone and being a male? He may live his entire life and not know he's a super male. That would probably mess with his ego. Well, if he found out later in life, 80 years old, and found out he's super male, man, I can see his pickup line. See, hey, babe, you know what's super male? Yeah. He may very well live his entire life and not know it. He normal intelligence, he can reproduce. Although both of these will run higher risk of possibly creating this or possibly this through non-disjunction, which I'm going to go back to in just a second. This person, there is one thing that might be involved with this person that would trigger a genetics test to figure this out. If this male is carrying a bald gene for baldness, this person will go bald at puberty. It won't be at 25 or 30 or 35 or 40. This person will start going bald at puberty, losing chunks of hair. So that's going to be kind of a heads up. You know, kid wakes up and there's hair on his pillow, comes back from school and he's missing some more. Over a short time frame, the kid's bald, and mom's like, what is going on? Takes him to the doctor. The doctor looks at all the symptoms, runs a genetics test, comes back, sits mom down and say, your son is a super male. She said, well, thank you. Thank you very much. He's a good boy. And then he goes on to explain that the problem with super male with extra testosterone, he has a much increased chance of liver cancer and testicular cancer. And it's really no different than a person taking steroids at a young age. Excessive steroids is what's happening. So what's a doctor going to suggest to possibly help fix this problem? Give him some estrogen? Let's turn him into a female. It would be nice if we could counteract some of that. He's actually going to suggest getting rid of some testosterone. How would you do that? I know you guys think of it, but you're not even going to say it. We're going to remove one testicle. That would do it, wouldn't it? So yeah, but he'll never be the same again. Well, rather be alive and not quite the same, or dead. There's some choices there, right? So if we remove one testicle, would we lower testosterone levels? Sure. Can this person still have a normal life? Could this person still reproduce? Yeah. Absolutely. I actually had one of these come through class. Wouldn't have known it except they told me. Once they told me, yeah, okay, you fit some of the symptoms. He was bald-headed. In fact, he carried an extremely sharp knife with him, and that's how he shaved his head. He just shaved his head. And he showed me, if he let his hair grow out, it would just been blotches. It would look really, really weird. But big guy, he could have been a linebacker somewhere. He did play a lot of high school. I mean, he is a big guy, but he loved science. He was very intelligent. He went through every class I taught him. When he left here, I don't remember where he went to college, somewhere down in Arkansas. He and his girlfriend went to that same college, got married within that next year. Within the next year, he was diagnosed with testicular cancer. They removed the one testicle that had the cancer, not both. And I know after that, I kept up with him for several years. I hadn't talked to him probably in 6 or 7 years, but they've had 2 children, and he's been in remission ever since. Now, what if as a teenager, of course we're just learning about this stuff when he was a teenager. What if as a teenager the doctor suggested removing one testicle? Would he have ever had cancer to start with? Chances are probably pretty slim, weren't they? Then I have this question. Students ask me, well, what if when he was younger and they removed one testicle, what if they took the wrong one? Think about that. It's a little bit of a silly question because the whole purpose is to prevent cancer to start with. In his case, he got lucky. He only had one with cancer, and the other one didn't. Now that was, but to prevent it up front is the best case scenario. So there's a case of super male. Now I will say a couple other things on this. Extra testosterone in these individuals, they tend to have anger issues. I never knew him to have an anger issue, but I didn't know him personally. I didn't spend a ton of time with him or anything like that. I will say this though. I hired him to do something out on the farm, and he come out I was going to show him what to do, and it's the first time he ever met my wife. Now, does any of y'all know my wife? I know you do. But he met my wife come out to tell me something, and probably bark orders at me, you know how wives are. I was going, yes ma'am, yes ma'am. And when she went back in, he looked at me and said, man, Mr. Dwann, you give us ugly folks lots of hope. I said, well, count yourself ugly, but I'm not ugly. So I did pretty good. Guys tend to marry up, girls tend to marry down. So that was her loss and my gain. So us ugly folks have hope. So now, a temper issue. Also, some psychologists were doing some research in a prison. Yeah, and in this prison, it's going through a lot of the folders and the crimes and things, and they found that 5% of the prisoners in this particular prison, it's all male prison, had this genetic problem. So they thought, They put together a hypothesis real fast. Their hypothesis was, these guys are in prison because of the extra testosterone. They're in here because of sex crimes. That's why they're in here. The testosterone drove them to sex crimes. So when they finished their research, they found no significant evidence to prove that hypothesis. In fact, it disproved the hypothesis and found that the other 95% of the population in the prison were equivalent percentage was in there for sex crimes, and the rest of them were robbing the 7-Eleven. You know, it was that kind of scenario. So these guys were robbing 7-Elevens, and a small percentage were in there for sex crimes. So that didn't hold up. But they did note in that anger issues. Almost every one of them with this configuration had notes in their file about anger issues. Okay? Now, I've given this lecture before and had students say, my husband's like that. It doesn't mean he has this. If he has an anger issue, maybe you should be nicer to him. Maybe you're driving to the anger issue, right? All right, so any questions there? Now, how does this happen? If we look at non-disjunction, let's look at, here we are in metaphase of meiosis. We have sister chromosomes lined up. All right? In anaphase of meiosis, those chromosomes are supposed to move to opposite poles, right? In non-disjunction, they don't. You might have this one right here stays together and all moves over there and nothing there. Okay? What if this is the one that's fertilized and we end up with an XO, a sperm that's XO? What do we have? Turner syndrome. What if a Y sperm, what do we have? Dead. You got to have an X to be alive. Nothing. That's what you have here. What if, this is the one, what if a Y sperm fertilized that one? What do we have? Klinefelters. What if an X sperm? What if we have non-disjunction in the male? Well, let's just drop down to this one here. What if we have non-disjunction in sperm and a double Y sperm connects that one? Yep. So non-disjunction is where sister chromosomes don't separate. They stay together. And then fertilization takes place with either a Y or an X. And you have meta-female, super-male, Klinefelters, or in some cases, Turners. So it makes sense? That's non-disjunction. All right. All right, I have one thing left, and this is actually in your notes. Yeah, I think I may have already done this. I already did cell respiration, didn't I?
Yes.
Yeah, it's actually in this chapter. And I'm glad I've already done it because I really didn't want to do it today. From that, I think we're ready to break into a lab. Does anybody have any particular question at this point? All right, I think I'll cut the video at this point.